Dear students, welcome you all. Today I am going to discuss vegetative propagation, artificial methods and their economic values. Vegetative propagation is the formation of new individuals from any vegetative part of the plant, including stem, root or leaf. The structural unit that is employed in place of seed is called as propagule. Vegetative propagation can be natural or artificial. Artificial methods of vegetative propagation can be achieved with different methods. The first method is the bud graft, in which a stem piece containing bud is inserted into a slot cut in a root stalk. The second method is the tissue culture technology, in which the single plant cells can be induced to divide. With the division of the cells, they grow and make a complete plant. Third method is the cutting, in which plants are vegetated by putting the cut ends of a shoot into water or moist soil. When they are provided with suitable conditions, the roots grow from the base of the stem into the soil, and the stems and leaves also appear. First, we will discuss natural vegetative propagation. Vegetative reproduction by natural method is very common among seed plants. Vegetative organs such as root, stems and leaves bear adventitious buds and bring about the formation of new plants. First, stems. Aerial weak stems, which include runners and stolons, when they touch the ground, give off adventitious roots. Thereafter, if connection breaks from the parent plant, the portion of newly formed roots develops into an independent plant. Next is the root. New plants grow out of swollen modified roots, which we called as tubers. Buds develop at the base of stem and then grow into a new plant. Leaves. Leaves of some plants grow into a new plant when they become detached from the parent plant. Other plants grow small plants called as plantlets on the edge of their leaves, for example, bryophyllum. Other vegetative organs which carry out natural method of propagation include bulbs, corms, tip layers, etc. Now we will discuss artificial method of vegetative propagation. Various methods of vegetative propagations have been developed by plant growers and horticulturists for commercial production of crops. These are called as artificial method of vegetative propagation. Now there are various methods of artificial vegetative propagations which are described as number first, cuttings. Many types of plants, both woody and herbaceous, are frequently propagated by cuttings. A cutting is a vegetative part which is severed from the parent plant in order to regenerate itself, thereby forming a whole new plant. New roots and leaves grow from the cutting. Take a cutting with a sharp blade to reduce injury to the parent plant. Dip the cutting tool in a rubbing alcohol or a mixture of 1 part of bleach to 9 parts of water to prevent transmitting disease from infected plant parts to the healthy ones. Remove flowers and flower buds to allow the cuttings to use its energy and stored carbohydrate for root and shoot formation rather than fruit and seed production. To hasten rooting, increase the number of roots by using a root hormone, preferably one containing a fungicide. After the proper root formation, the whole cutting will develop into a new plant. Now, cuttings may be of various types, as stem cutting. Numerous plant species are propagated by stem cuttings. Most of them can be taken throughout summer and fall, but stem cuttings of some woody plants root better if taken in fall or in the dormant season. Success with herbaceous plants is generally enhanced when done in the spring. These plants are actively growing then and more apt to root quickly on their own. There are several different types of stem cuttings depending on the part of the stem needed. At least one node should be below the media surface, although some plant root at internodes, others only root at nodal tissue. Another method which is called as medial cuttings or stem section cuttings. In this, the first cut is made just above a node and the second cut just below a node 2 to 6 inches down the stem. The cut is then prepared and inserted as in case of tip cutting. Care is needed for the position of the cut ends, that is, right side up and base down. Another method is called as can cuttings. In this, can-like stems are cut into sections containing 1 or 2 eyes or nodes. Cut ends are dusted with fungicide or activated charcoal and allowed to dry for several hours. About half of the cutting is laid horizontally below the media surface, eye facing upwards. Can cuttings are usually potted when roots and new shoots appear, but new shoots from plants like Dracena and Croton are often cut off and develop roots in sand. Single eye method. The eye refers to the bud which emerges at the axil of the leaf at each node. This is used for plants with alternate leaves when space or stock materials are limited. The stem is cut about ½ inch above and ½ inch below a node. The cutting is placed horizontally or vertically in the medium with the node just touching the surface. Double eye method. This is used for plants with opposite leaves and space or stock material is limited. The stem is cut about ½ inch above and ½ inch below the same node. The cutting is inserted vertically in the medium with the node just touching the surface. Heel cutting method. This method uses stock material with woody stems efficiently. A shield-shaped cut is made about halfway through the wood around the leaf and axil bud. The shield is inserted horizontally into the medium so that it is completely covered. An leaf blade is removed, but a portion of the petiole is kept intact for ease in handling the small cutting. Leaf cuttings. Many plants can be artificially cloned by leaf cuttings. Species that work well include those of Bryophyllum, Crassula, etc. In this process, a leaf containing blade and petiole is removed from the donor plant. The leaf is placed in moist soil or in water and kept in light. Now, propagation through leaf can be achieved with the following methods. First is whole leaf with petiole. The leaf with petiole up to 1½ inch is detached and inserted its lower end into the medium. One or more new plants form at the base of the petiole. The leaf may be severed from the new plants when they have their own roots, and the petiole can be reused. Another method is whole leaf without petiole. This is used for plants with sessile leaves, that means with no stalk or petiole. The cutting is inserted vertically into the medium. A new plant forms from the axillary bud. The leaf may be removed when the new plant develops its own roots. Another method is split vein method. A leaf from the stalk plant is detached and slits are made on its veins on the lower surface. The lower side of the cutting is laid down on the medium. New plants form at each cut. If the leaf tends to curl up, it can be held in place by covering the margins with the rooting medium, and the examples include Rox begonia, as you can see in the figure. Another method, which is called as leaf sections. This method is frequently used with the snake plant and fibrous rooted begonias. Begonia leaves are cut into wedges with at least one vein and flayed down on the medium. A new plant arises at the vein. In snake plants, leaves, 2 each sections are made. Consequently, the lower cut is made slanted and the upper cut straight. The cutting is then inserted vertically, roots develop, and eventually a new plant appears at the base of the cutting. It is to be noted that with variegated snake plants, the new shoots develop from cells that do not display the variegation. Root cuttings method. Root cuttings are usually taken from 2 to 3 year old plants during their dormant season when they have large carbohydrate supply. Root cuttings of some species produce new shoots, which then form their own root system, while those of other plants develop root systems before producing new shoots. Another method, plants with large roots. A straight top cut is made, then a slanted cut is made 2 to 6 inches below the first cut and stored about 3 weeks in moist sawdust, peat moss or sand at 40° Fahrenheit. The cutting is removed from storage and inserted with the top approximately level with the surface of the rooting medium. This method is often used outdoor plants. Plants with small roots. 1 to 2-inch sections of roots are taken and inserted horizontally about ½ inch below the medium surface. This method is usually used for indoor plants. Now another method is grafting, which is a very important method. In this, two plants are used to develop a new plant with combined traits from the two parent plants. Here, the scion and stalks are used. Scion is the above ground part of the plant, while the stalk is the rooted part of the second plant. The scion is inserted into the cleft made in the stalk. Layering is another important method. In layering, a shoot of a parent plant is bent until it can be covered by soil. The tip layer of the shoot remains above ground, which results the formation of new roots and eventually a new plant. These plants are then separated. This method of vegetative propagation promotes a high success rate because it prevents the water stress and carbohydrate shortage that plug cuttings. Some plants layer themselves naturally, but sometimes the plant's propagators assist the process. Layering may be enhanced by wounding one side of the stem or by bending it very sharply. The rooting medium provides aeration and a constant supply of moisture. Now layering can be of following types. Number first, tip layering. In this method, a hole 3 to 4 inches deep is dug and shoot tip is inserted and covered with the soil. The tip grows downwards first, then bends sharply and grows upwards. Roots develop at the bend and the recurved tip becomes a new plant. The tip layer is then removed and planted in early spring or late fall. Examples include purple and black raspberries or trailing blackberries. Another method, simple layering. The stem is bent to the ground and covered part of it with soil, leaving the last 6 to 12 inches exposed. The tip is bent into the vertical position and stalked in place. The sharp bend often induces rooting, but wounding the lower side of the branch or loosening the bark by twisting the stem may help. Another method, compound layering. This method works for plants with flexible stems. The stem is bent to the rooting medium and for the simple layering, but alternately stem sections are covered and exposed. Another method, mound or stool layering. In this method, the plant is cut back to 1 inch above the ground in the dormant season. The emergent shoots are mounded with the soil in the spring to enhance their rooting, and the examples include gooseberries, apple stalks, etc. Another method is called as air layering. Air layering is used to propagate some indoor plants with thick stems or to rejuvenate them when they become leggy. The slit is made just below a node. The slit is opened with a toothpick and the wound is surrounded with wet, unmilled sphagnum moss. The sphagnum moss is wrapped around with plastic foil. When roots pervaded the moss, a cut is made below the root ball, which results the new rooted plant. Now, another important tool which we called as tissue culture. To make thousands of individuals of one clone, special method is used called as plant tissue culture or micropropagation. Micropropagation, also called as in vitro clonal propagation, is the multiplication of genetically identical individuals by asexual reproduction. In this process, the genetic stability of new shoots is dependent upon their origin. This is also a kind of rapid vegetative propagation, and hence the products of this process should be considered as a single clone. The significant advantage of clonal propagation over the conventional method is that in a relatively short span of time and space, a large number of plants can be produced starting from a single individual. It has been estimated that axillary bud proliferation approaches typically results an average 10-fold increase in shoot number per monthly culture passage. Now, the procedure of plant tissue culture. First is needed donor plant or donor tissue. This is called the explant. The tissue explant has to be at least parenchymatous, but it is better if it is meristematic. It has to be first surface sterilized. This is done by immersing the tissue in a solution of sodium hypochlorite, rinsing in sterile distilled water, and then slicing off the killed surface layers. Number second, The explant is placed in a sterilized liquid medium on a shaking machine. This medium contains water, minerals, vitamins, sugars, and dilute oxygens, usually indole butyric acid. The oxygen stimulates the cell of the explant to divide rapidly to form the callus. The callus is broken up by shaking action. Thus, the liquid medium becomes loaded with small callus pieces. And number third step, the callus pieces are now spread across sterile solid medium. The medium contains water, minerals, vitamins, sugars, and cytokinins. The medium is solidified into a gel by adding a small amount of agar. The container is kept under light. The combination of cytokinin and light causes shoots to be initiated on each callus piece. And number fourth step, the shoots are moved into a solid medium that contains water, minerals, and agar. Depending on the species, there may also be some oxygen in the medium. The oxygen from the leaves or in combination with the oxygen from the medium causes root initiation. And number fifth, the plants can be moved to soil and slowly eased from humid conditions to successively dried conditions. Ultimately, the plants can be moved to field conditions. Thus, the following stages are involved in micropropagation. Stage first, which is called as establishment of aseptic culture. In this stage, culture is initiated which is free from infection. It includes cultures like shoot tip, stem, etc. after proper sterilization process. Stage second, multiplication of propagules. The purpose of this stage is rapid multiplication of propagules or cultures that eventually produces plantlets continuously. Mostly this is obtained by axillary shoot initiation or adventitious organ formation. Adventitious shoot formation is preferred since it utilizes the somatic or asexual embryo genetic pathway, which is much faster. However, in this process, plant regeneration is achieved at intervening callusing stage so that there may be high risk of genetic variability in plants. While in axillary shoot multiplication, this incidence is almost absent, but the rate is much slower. Stage third, root induction and preparation prior to transfer. This involves the preparation of propagules for their transfer from in vitro to in vivo stage. The process primarily involves the root induction and hardening of the plant. Although root regeneration has been done in vitro conditions, there is a growing interest to root them in vivo conditions, and it has been reported successfully in many cases. Stage fourth is the transfer to field or outdoor. The successful transfer of in vitro grown plants to the field with the high survival rate is the main aim of this stage. To improve the survival rate, a number of methods have been applied, particularly under greenhouse conditions, which includes the use of mist, carbon dioxide enrichment, optimization of light and temperature, etc. Stage 0, preparatory stage of the mother plant. Recently, this stage is proposed to prepare the mother plant to be used as a source. In this stage, the mother plant is growing strictly under hygienic conditions. Mother stock plants are treated with plant growth regulators such as BAP to improve the success in stage first. Also, the application of growth retardants is not uncommon, despite other recommendations to prepare a mother's plant at stage 0. The actual practice is limited. A variety of chemicals are also used which include medium plant growth regulators, carbohydrates, etc., and physical factors like light, temperature, pH, etc. to regulate in vitro responses. Now, there are a number of advantages of vegetative propagations which include the offsprings are genetically identical and therefore advantageous traits can be preserved. Only one parent is required, which eliminates the need of spatial mechanisms such as pollination. It is faster and helps the organism to increase in number at a rapid rate that balances the loss in number due to various causes. Many plants are able to tide over unfavorable conditions. This is because of the presence of organs of asexual reproduction like the tubers, corms, bulbs, etc. Vegetative propagation is especially beneficial to the agriculturists and horticulturists. They can raise crops like bananas, sugarcane, potato, etc. that do not produce viable seeds. The seedless varieties of fruits are also a result of vegetative propagation. The modern technique of tissue culture can be used to grow virus-free or disease-free plants. Availability or regeneration of plant throughout the year, that is, in all seasons. Resistance of plants to insects, disease and herbicides. Next is the production of uniform clone from highly heterozygous plants. Mass production of plant cells and tissues as a source of secondary metabolites. Dear students, we come to the end of the topic artificial methods of vegetative propagation and its number of types. We discussed that these methods are very helpful for crop improvement and preservation of medicinal and aromatic plants. With this, I take your leave. Thank you.